Hello? Oh, there we go. Good. Hello, everyone. Good afternoon, and welcome to this year's Naimark lecture. My name is Jim Butler, and I'm the Associate Dean for Undergraduate Medical Education here at the Max. Naimark lecture is annually given since 2013 and is sponsored by the class of 1972 in honor of Dr. Arnold Naimark. Today, to give a lecture on crafting policy on transplantation, the challenge of reconciling science and public expectations, is being delivered by Dr. Peter Nickerson, who is Dean of the College of Medicine, as well as Dean of the Faculty of Health Sciences here at the Rady Faculty of Health Sciences. I'd like to welcome Dr. Nickerson, who has published over 190 research papers, reports, reviews, and book chapters, and has extensive research experience in nephrology as well as in transplantation, and a significant clinical career as well as research. Welcome, Dr. Nickerson.
Thanks, Jim, and also want to acknowledge Dr. Naimark, who's this lecture is in sponsorship for. I was going to start by acknowledging the University of Manitoba campuses are located on the original lands, the Anishinaabe, the Ininewuk, the Assiniboine, the Dakota, the Nene, and the Inuit, and on the national homeland of the Red River Métis. We respect the treaties that were made on these territories, and we acknowledge the harms and mistakes of the past, and we dedicate ourselves to move forward in partnership with Indigenous communities in a spirit of reconciliation and collaboration. And part of that is trying to, as we continue to look at expansion in the medical school, developing pathways for Indigenous students from high school through undergrad and then into med school. And it's a priority that we have now in our strategic plan, and I look forward to trying executing that over the next few years. So I was asked today to talk about crafting policy on transplantation and the challenge of how we reconcile what we know about the science of transplantation versus what the public expect to get through organ transplantation. And this has actually been a journey that's been going on for over 100 years. We're going to talk about the history of kidney transplantation going back to the 1900s, where Manitoba has contributed over the last 50 years or so, and then what is health policy and can it meet public expectations in this area, and what might be future directions where we see integration of both science and health policy moving forward. So to start with the history, this goes back to 1902 in Lyon, where Alexis Carrel, a surgeon who was working in France for a while and then moved to Chicago at one point, he described how to actually anastomose an artery to another artery. So an end-to-end anastomosis. And without this surgical technique, you cannot do organ transplantation. So he described this back in 1902, and he was given the Nobel Prize in 1912 for this technique, which is still utilized today in vascular surgery overall. So from the 1900s to the 1930s, we started trying with this surgical technique to do transplantations, and first it was animal kidneys to humans. In 1906, there were attempts at pig kidneys or goat kidneys, and then monkey kidneys. And these all failed. And it's kind of interesting because when you read the paper nowadays, in 2025, you're hearing about pig transplants in humans again, where it's taken us over 100 years to figure out whether we're closer to a solution of using pig kidneys instead of having to use human kidneys. But all of these initial transplants failed, and they failed pretty spectacularly, and we learned that there's a problem of species compatibility between other animals and humans that will present a major barrier to a successful transplant. So the first successful transplants actually were used in burn patients, where they could take human skin grafts to take skin from deceased individuals and put it on burn patients to protect that burn area while there was healing going on, and eventually that skin would be rejected because the immune system would recognize it as not compatible. And then when they tried to use skin a second time on a skin graft, if it was from the same donor, it would actually be rejected quicker, and that's where we learned about immune memory, that the immune system, once it's seen a foreign tissue, will react much more aggressively the second time around. The story then goes on to 1945 to 1955, where they were doing then a human-to-human, a bridge-to-life kidney transplant. And this is a case in 1947 where a patient was in a coma because they had sustained acute kidney injury, so their metabolism was off and they were uremic, and so they were encephalopathic. And they put a deceased donor kidney into this individual. The kidney only worked for 2 days, but that 2 days was long enough for their own kidneys to recover from their acute injury. And so this patient woke up and survived because their native kidney actually started to function again. There was no dialysis in the 1940s. Dialysis doesn't show up until the 1960s. Then the next group that tried to actually do some kidney transplants was in Boston. And they did 9 kidney transplants, and they all failed spectacularly. And again, they reaffirmed that tissue compatibility is a major barrier to crossing the immune system. So that took us then to trying to decipher what is the immune system involved with transplantation. And Peter Medawar was an immunologist in London. And this is the sign that's on his home in England, where he actually was doing animal experiments looking at tissue compatibility and looking under the microscope at these rejected organs and realizing that there was lymphocytes, or what we call today T cells, and antibodies that were causing a humoral response that were attacking the graft. And for this, he won the Nobel Prize in 1960 for describing the immune response to transplanted tissue. The 9 transplants that were done in Boston were done by David Hume, and what David was doing when these kidneys were being lost was he was actually recovering the kidneys and the lymph nodes that were draining these kidney transplants and examining the pathology under a microscope. And he actually went on to describe what Medawar had been seeing in the animal model systems. He was describing now in the human kidney grafts, and he came up with this cartoon suggesting that the lymphocytes and the antigens were coming out of the kidney, were moving to the regional lymph node, activating the immune system, and that this was leading to an immune response that came back in through the artery to the kidney that would lead to rejection through both antibodies and T lymphocytes. And so this was really the first description of the histology of rejection that was taking place in human transplants that were all failing. As I said, this was 1955. Well, the first successful kidney transplant happened in Boston, and it happened in the 23rd of December 1954. And the surgeon Joseph Murray and John Merrill and Hartwell Harrison were the other physicians involved. They did identical twin transplants. And they had reasoned that if they were genetically identical twins, then they should be able to get a transplant to go from one to the other without ever causing a problem in terms of activating the immune system. And so this was the event. And what they did to prove that these truly were identical twins is they took what they'd been learning by skin grafts, and they did these skin grafts here where they took the skin of the twin and transplanted it onto the other recipient and looked to make sure that it wasn't rejected. And if it wasn't rejected, and there's no drugs here, then they would assume that they must be genetically identical. Otherwise, you'd expect the graft to be lost. So that was true, and that started off transplantation. And actually, there were some Winnipeg identical twins that went down to Boston and had some transplants in those early days. And that was really all we had to offer, until we had immunosuppression. And Joe Murray, the fellow that was doing the twin transplants in Boston, he started using radiation therapy, 6-mercaptopurine and prednisone, and he was radiating the whole lymphoid system, thinking that that would be immunosuppressive and allow the organ then to be accepted. And the success of that approach was that 40% of the recipients of a kidney transplant would still have that kidney surviving at 1 year, and 80% were seeing rejection of the kidney by a year. The next major advance was a fellow from Cambridge named Roy Kahn, and he came over to Boston and worked with Joe Murray. And they had a drug called Imuran, which is azathioprine, which inhibits cell proliferation by inhibiting DNA synthesis. And that allowed them to get to 1-year graft survival rates of 65% and rejection rates of 60%. And then the next major advance was the development of the drug called cyclosporin, which is a calcineurin inhibitor, which inhibits T cell activation. And this was discovered by J.F. Borrell, who had been in Norway digging up soil samples and found fungi. And from the extracts of the fungi, he was incubating them with lymphocytes and showing that they could inhibit the lymphocytes in vitro in a test tube. And on the basis of that, they developed cyclosporin. And once they had that, that got us into the modern era of transplant, where we had 1-year graft survival rates up to 90% and rejection rates down to 45%. So if you go up to 1963, in the whole world, there had only been 244 transplants done since the beginning of the 50s. As of 2021, we're doing globally over 150,000 transplants a year in the world. And this is really the drug combination that we're using, prednisone, antimetabolite, and a calcineurin inhibitor. Now, the next major story that was happening in transplant was this fellow, Paul Terasaki. He was a biologist by training, and he went and worked with Medawar in England. He had actually was Japanese, living in the U.S., and during the Second World War, he'd been actually in the camps, interned. And after that, he went to UCLA, did his biology degree, then went off to England to study with Medawar, and came back to UCLA. And he invented this little plastic tray, and he patented it. And that got him inducted into the National Inventors Hall of Fame in 2018 for making this tray. And what he did in this tray was he took donor cells and he put serum from the recipient and he threw in complement. And if the cells got killed, that was called a positive cross-match. So they would read out in this little micro well whether there was cell death or not. Up to this point, when they were doing kidney transplants, you basically took the donor and the recipient, and you put the kidney into the recipient, you gave them immunosuppression and hoped for the best. What he did was a retrospective study and said, had I done a cross-match with this technique in the serum that was to detect antibodies that the person might have had preformed, and it was positive, causing cell death, 24 of these 30 transplants with a positive cross-match, they all lost their graft within 2 weeks. If they had a negative cross-match, the majority of them kept those kidneys functioning out towards a year. And so with this technique, this became a contraindication of transplant. Nobody could go to a transplant without a prospective cross-match to make sure that the recipient didn't have memory towards the donor's tissue. And you might say, well, why would they have memory? Well, if you're a woman who has kidney failure and you have had pregnancies and your husband's the organ donor for you, you've been potentially sensitized to your husband through the fetus, because the fetus has one chromosome that's coming from the father and the other chromosomes coming from the mother. And so in utero, you get exposed to the father's HLA, or tissue type. And that then could lead to sensitization. And so you could have preformed antibodies in your blood against the potential donor. The interesting thing about this paper, this was published in 1969. Paul was 40. He, on the basis of this, formed a company, and he sold the company a few years ago for $1 billion. So he basically, with this patent for a little plastic tray, built a whole industry on doing tissue typing pre-transplant and developed some of the best tools that we all have nowadays that are used globally for testing compatibility. So life begins at 40. It doesn't begin in the 20s. The next major step was Jean Dosset, and again, a French immunologist And he was studying how viruses were being detected by the immune system. And what he discovered was the HLA genes that are on chromosome 6. And what we've realized now is that these are the major histocompatibility genes. These gene products, which are the genes that present antigens to activate your immune system, are what are foreign when it's a donor's HLA that's not the same as your HLA. And what we know is that if you have a 0 HLA DR A and B mismatch, the graft survival on average is about 12.6 years, but if you have a single A, B, or DR mismatch, the graft survival goes down by 4 years overall. So histocompatibility and matching, the more similar you are to your donor, the more likely you are to keep that kidney after a transplant with the drugs that we give you. The next major advent since 2000 is where we are today. Imuran's been replaced by mycophenolate or Cellcept. It's a more effective cell cycle inhibitor. And tacrolimus has replaced cyclosporin as a more effective calcineurin inhibitor. And now our rejection rates are down to 15%, and our graft survivals are up to 96% at 1 year. The other thing people started to appreciate was that the quality of the donor organ itself also was correlating with how well these transplants would do. So a lot of things go into a deceased donor's consideration. What's the donor age? Did they have a history of hypertension or diabetes? What was the cause of death? How well was their kidney functioning when they died? And was it a neurologic death or was it a cardiac death? And if it's a cardiac death, you're more likely to have some ischemic injury to the kidney before you're able to get it out of the donor. One of the biggest correlates of this kidney donor profile index, or KDPI, is the donor age. So as you're young, if you're very young, you have a high KDPI. It goes down to when you're about 15 to 19. That's like the ideal donor. And as you get older, your KDPI index starts going up. And what that translates into is that a low KDPI, let's say 1 to 5%, these have 70 to 72% 8-year graft survivals, whereas a KDPI that's in the 90 to 99%, their 8-year graft survivals are down around 25 and 30%. So a higher quality organ is going to survive longer in a recipient. So that's sort of some of the background of what got us to where we are today. Whereas Manitoba had some contributions here, well, we celebrated our 50th anniversary in 2020 in the middle of the pandemic. The individuals that started transplant in Manitoba were Al Downs, a vascular surgeon who I think is probably one of the best vascular surgeons we've ever had. Although I think the other vascular surgeons of today might argue with that. I still think Al was one of the best vascular surgeons that I ever had encountered. And Ashley Thompson was a nephrologist, was considered the grandfather of nephrology and dialysis in Manitoba. And we went on then to have a whole transplant program led by John Jeffries, who came from McGill, David Rush, who came from Argentina, Rachel McKenna, who came from Ireland, and Kim, who was our nurse manager of the transplant program for many years. And we opened up in the middle of the pandemic our new transplant wellness center, which I think is one of the nicer centers in the hospital today. So some of our innovations was we wanted to try and improve the cross-match technique. The CDC cross-match, if you started diluting out a serum that you knew had antibodies against HLA, it would stay positive down to about a 1 in 2 dilution. But with a flow cytometer, you can now look at fluorescence on the cell surface rather than cell death. And with that, we were able to detect weak antibodies down to a 1 in 64 dilution. And what we were able to show was that if you use flow cross-matching, it actually predicted those individuals who are more likely to have early graft loss compared to a negative flow cross-match and have more early rejections and more severe rejections in the early post-transplant period. So Manitoba was one of the first centers in Canada to move to a routine flow cross-matching to mitigate the risk of losing kidneys early from this low level of immune memory being detected. David Rush, he actually was a very innovative individual when he started out here in the 1980s. And through the 90s, you know, most people in transplant thought, well, if your kidney function was stable, it meant that you didn't have any rejection. And David challenged that whole paradigm. What he did was he did biopsies in well-functioning kidneys to ask the question, is there rejection when you don't think there's rejection. And what he found was there was a lot of subclinical rejection in about 30 to 45% of these biopsies in the first year when you were on cyclosporin, Imuran, and prednisone. This led to him doing a randomized control trial to say, okay, if I treat these subclinical rejections, do I have a better outcome? And he was able to show that those that were treated actually had better creatinines at 2 years compared to those that did not get protocol biopsies. And then he went on to a multi-center Canadian trial showing that with current immunosuppression, it actually did better at decreasing the rates of subclinical rejection. And this whole body of work led to David getting the Order of Canada for his work in transplantation. More recently, what we've shown is not just biopsies early when the kidney's functioning well to see if there's rejection then, but what about if you've treated somebody with rejection? Does the rejection go away when the creatinine is stable? And so we did a series of studies this is Dr. Chris Wiebe, who many of you may be meeting through the nephrology course. And what he showed was that 37% of patients have persistent rejection even after we treated them and thought we had treated effectively for rejection. And what he showed is that this correlated with increased risk of graft loss. And Dr. Julie Ho in our program, in a systematic review, also found that in the literature, 39% of patients would have persistent rejection 2 to 9 months after a rejection therapy. And this is really talking about the lack of clinical trials that even to today are not in place to define what is optimal anti-rejection therapy. And it's a trial that now, between Winnipeg and Australia, we're trying to work out what's the best way of treating rejection. And there's been no area of development in this over the last 40 years. The last paper looking at treatment of rejection goes back to the 1980s. The other innovation we had was not just doing biopsies to surveil the grafts, but actually monitoring for the development of antibodies against the graft over time. And nobody was doing this. Everybody thought, well, if the kidney functions fine, you don't need to look for anything until there's a problem. And what we found was that if you could actually detect an antibody in the blood post-transplant, this correlated with a marked decrease in graft survival. And we showed that that correlated with the onset of the antibody leading to a rapid decline in kidney function. It was related to antibody-mediated rejection occurring in the graft. So Winnipeg's been actually at the forefront of really defining how to monitor for rejection and decide whether you're effectively being treated. And it leads to this sort of understanding now of what's leading to graft failure or death of an individual after a transplant is the loss of organ function because of fibrosis in the kidney or fibrosis inside the glomeruli. This can be recurrent disease. We see this with IgA nephropathy. But most of it is driven by either a memory immune response or the degree of mismatching between the donor and the recipient for the HLA molecules that's leading to T-cell mediated rejection that could be subclinical or clinical, or development of a donor-specific antibody that leads to antibody-mediated rejection that again can be subclinical or clinical. And overall, you get what's called a smoldering rejection in these grafts that slowly, over time, you start losing kidney function. Donor age and ischemia reperfusion injury can lead to delayed graft function that can lead to scarring. And what we do then is we give drugs to try and suppress the immune response from developing, but they themselves come with side effects, metabolic dysfunction, infections, or risk of cancer, or risk of drug toxicity in the kidney, or the development of viral infections in the kidney. And so when these things start happening, then we tend to minimize the drugs that we're giving, or the patients don't like the side effects, so they stop taking the drugs that we're giving. And this leads to then taking the brakes off of rejection, which can lead to graft failure. Okay, so that's sort of the science behind where we're at. What about, where's health policy fit into this? Well, why do we do transplant versus dialysis? Well, if you look at the patients who are on the ready list to be transplanted but are not transplanted and stay on dialysis, the mean survival, and then looking here at age 45 to 65 in the blue and the gold, the mean survival on dialysis is about 8 years. If you have a transplant, your mean survival is going up to about 16 years. So you're basically doubling life expectancy with a transplanted organ than being on dialysis. You have better quality of life, and there's certainly a cost benefit. It's a lot cheaper to have a patient with a transplant and the cost of the drugs versus the cost of running a dialysis program to maintain these people. So where are we at in Manitoba? Well, we have 1,800 people on dialysis. We have 500 of them that are in the process of being worked up or are ready to be transplanted, and we're transplanting 70 to 80 per year. So the real question becomes then, who gets the kidney when there's a kidney available? When you look at organ donation, only 3% of in-hospital deaths are ultimately eligible for organ donation. And there's been a lot of debate, do you need mandatory referral policies or do you need a presumed consent policy? A presumed consent policy says when you die, you're automatically going to be considered an organ donor in the province. We don't have that. What we have is a mandatory referral policy. Once you die, before you have to be considered for organ donation. And in both of these cases, we talk to the family and we ask for consent of the family. And what's really important is, do the family know what you want? If the family doesn't know, only about half of the time will they give consent to proceed to organ donation. If they know that you want to be an organ donor, it'll be over 90%. So what your family knows about what your intentions are is paramount to whether you're ultimately going to be potentially an organ donor. We have transplant coordinators and ICU physicians that assess donor eligibility, looking at the organ quality and the potential for a donor-derived disease that would prohibit them from being an organ donor. And so in 2015, we introduced a mandatory referral policy. So here's public policy trying to drive up the number of organ transplants that we can do, given how many people are on the wait list. When we looked in 2015, before we introduced the policy, referred was 131, not referred was 287. So our routine notification of potential organ donor consideration was only at 31%. But as we kept pushing on the policy going forward, we're now up over 94% being referred for consideration of organ donation. So the policy itself is driving up the number of potential organ donors to be considered. So what happened in this 1 year, in 2020, we had 391 through an audit that we said could have been referred. Of that, 362 were referred. 86 were identified of this 362 as potential donors, of which only 27 did the family consent for consideration, and only 20 went on to become actual donors in this process. 32 of the families declined, and the large reason for declining, again, is they don't know what your intentions were. And then 27, there were system barriers, or they were unsuitable to going forward for donation. So what is the expectation of patients who are on the wait list? And what do we consider about who actually gets these 60 to 70 kidneys that are available in a given year? Some of it comes down to medical need. And most people agree that you should have access to ensure health and life. The people who really are in urgent medical need for a kidney are the ones that you're not able to dialyze. So if there's a problem with your vascular access or your peritoneum for peritoneal dialysis, if you're not able to dialyze somebody, then these people go right up to the top of the list for consideration of the next available organ to get transplanted. And the other priority we give is to children. We know that neurodevelopmentally and for growth, a child with kidney disease really needs to get a transplant if they're going to grow and develop neurologically at milestones. But then the other things that come into play are utility. What's the optimal use of the organ? So there's only so many to go around. Can we afford to not have these organs last as long as possible? And so how do we do that? Well, we avoid memory, so we do the cross-matching to make sure that we're not putting an organ into somebody who has memory, which we can't control, and you're going to lose that graft right away. The other one that comes into play is HLA match, and we'll talk about that in a second. The other is the high-quality kidney. We know that a low KDPI or a high-quality kidney is going to last longer, so who gets that kidney? And what's the most effective drugs? Well, we know it's TAC and MMF is the most effective combination, so those are the drugs that we give everybody. The other side is, what's the justice here? What's equitable access to organs? There's those individuals who have immunologic memory. As I told you, women who have had pregnancies can develop HLA antibodies, and they can become broadly sensitized to all the HLA molecules in the population. And once you get your PRA above 80%, that means you react to 80% of all donors. Only 20% of donors are going to be compatible to you. And then they're also going to have to be blood group compatible. So women who have had pregnancies are disadvantaged if they're sensitized to getting access to a kidney transplant. Same is true of a blood transfusion. If you get a blood transfusion, you get exposed to the HLA molecules of the blood donor. And even though we look after the blood supply and we filter the blood, you can't exclude HLA molecules from sensitizing the individual after a transplant. So individuals who've had a blood transfusion, for whatever medical reason, if they ultimately need a kidney transplant, they may be very hard to find a kidney for. So what priority should they get? And then what about HLA genetic diversity in populations? One of the problems we have is the majority of the donors are Caucasian. But we know that 50% of our population on the dialysis wait list looking for a kidney are not Caucasian. So if we put a lot of weight on HLA matching, we're creating ethnic and racial disparity in access to transplant. So this is a real issue. We know it's scientifically the best thing to do to get the longest use of that kidney, but we're actually not giving equity of access. It's not a just approach. What about the age of the recipient? If you have older individuals on the list, do they get access to a kidney when you have a young kidney available? Or should you be giving the young kidney to a young person? Where's the justice here and what policy should we develop? And then time on dialysis. Hey, I waited my time. I waited 6 years. Do I get my turn now? Or does a person with 2 years of wait time get the kidney ahead of me? So where's the justice in that? And then if I live close to the center, hey, I can get you into the hospital in an hour versus I got to fly you down from the north. Am I going to be against you in terms of who's going to get access to the kidney? So these are all parts of the equation that go into how do we decide who gets the kidney? And then ultimately, what's the transparency? What are the rules? What's our allocation policy? And what's the accountability? Did the program follow the policy? This may sound very obvious, but we didn't have a policy here before 2009 where we actually gave very rigid rules for who got access in what rank order. And that was really on the basis of we're clinicians. We know how to make good decisions. We're going to decide from the list who we think is the best recipient for this organ. That was true in the whole country. We moved, actually it was 2004, we moved in 2004 and said that's not good enough. Patients are saying, why don't I get the next kidney? They want to know, where am I on that list? So we moved to a fairly rigid process. And the way we developed our policy was we actually met with focus groups. We met with people who were on dialysis to ask them, what did they view as important and what were they willing to trade off in terms of getting access to a kidney? So everybody agreed, what we met with, and we had multiple focus groups. And the focus groups weren't run by us, the physicians. We had independent third parties running the focus groups because we didn't want to be biasing them based on what the physicians were saying. What everybody agreed to is that medically urgent, those who could not be dialyzed should be right at the top of the list and they should get the next kidney. If you're highly sensitized, you're really hard to find a match for, and you get that one shot, a needle in a haystack, you should get that offer above others. And if you're a child, and the pediatricians don't like donors in going into children less than 6 years of age because the vessels on the small kidneys are too small for the children, and they're more at risk to developing a clot. So they like a little bit older organs. But they want high quality organs going into the children because they want those kidneys to last as long as possible. And so what we came up with was a system where we looked at the KDPI and we said young kidneys should go into younger people, younger being less than 60. I no longer qualify as young. Those other KDPIs, 20 to 59, they should go to everybody on the list, and the older kidneys should be given to the older individuals. And there's actually data to say that giving an older kidney to a younger individual actually puts them at risk, so that you should to do this. You should confine your higher KDPI kidneys to the older recipients. Now, when we asked the patients about this, what they really cared about was that they had equitable access. So if I'm over the age of 60, that makes up a third of my wait list, I should get access to a third of all kidneys that are available. That's what they cared about. They didn't care if they got the younger kidney or they got the older kidney. They just wanted to know they got access to what they were in proportion to the wait list. And the same was true of our Indigenous recipients. We have 20 to 30% of our wait list being Indigenous. They should have access to 30% of all donor organs that are available. So when we went to this system of a rigid approach, we gave points for wait time. We gave a very few points for tissue compatibility. We didn't want to create disparity based on the HLA genetics, and then the degree of sensitization, how sensitized were you? And that then gives us a very ranked order, and the clinicians have to follow that rank order. They're not allowed to deviate from that unless there's a really good reason, and there's a very few numbers of reasons why you could do that. And at first, everybody didn't like the idea of the rigidity of it, but now everybody loves it because it actually takes that whole thinking that you know better out of the equation, and you actually start creating equitable access to organs for everybody. And then what we put in place, again, the first in the country, was an allocation review committee. It was chaired by an ethicist. We had administrators from the dialysis and transplant program, waitlist patients, transplant recipients, the hospital advocate, and a social worker. They made up the review committee. There was no physicians on the review committee. The physicians had to present to the review committee and confirm that we followed the policy. And if we didn't follow the policy, what the committee could do is if we were deemed egregious in doing that, then they could report us to the college as being offside. So that could come with significant discipline. So it was really a way of building trust in the system that we're, through policy, now giving equitable access, giving access that's marrying up both the science and what patients are expecting. And it's led to us to continue to grow the program. So this is the number of functioning transplants we have in Manitoba, continuously growing, especially since we put in the mandatory referral policy where everybody's getting moved forward. And we know, and we do regular audit, that the recipients receive kidney transplants in proportion to the waitlist demographics related to age, Indigenous, rural, that there is no discrimination now as to who's getting access because we're following a very rigid point system. And we've seen stabilization in the number of patients on dialysis. We're growing our transplant program and we're maintaining a stability in the numbers on dialysis. So this is our goal. Our goal is to continue to grow transplant, and if anything, we'd ideally see a shrinking of the number of dialysis recipients, especially if we can more effectively prevent and treat kidney disease. We should see that going down. This comes as a team. This is not just physicians. There's a whole host of individuals from all professions who are involved with the supporting the transplant program. But the next challenge is, that's what we can do in Manitoba. What about access challenges that are related to the Canadian Constitution? So the provinces have jurisdiction for health, and they protect it. There's 13 independent healthcare systems, kidney transplantations, all provinces have programs, and all provinces have a major burden of dialysis costs. And so if you're a province, you want every kidney that's available in your province to be used in your province because it's going to save. We're spending well over $100 million a year on dialysis. Anything that we can do to minimize that expense is money that we could be spending elsewhere in the healthcare system. So provinces don't want to share kidneys between provinces. 10 minutes, okay, I'll speed up. But what about equitable access for vulnerable populations? So these individuals who are highly sensitized, who are largely women who have had pregnancies, individuals who've had blood transfusions. How do they get access, especially if you're in a small province with a small donor pool? So there's 1,500 donor organs per year in the country. Manitoba has 20 donors. So your chances of hitting the jackpot if you're highly sensitized by being in a small province is actually not good. So what we did was we said, well, let's look at, can we start to develop a foundation for which across the country we could develop a sharing system for kidneys? And we went back to the flow cross-matching that we developed in Manitoba, which had come out of the US, but not that many programs were using it in the US. We said, this should be the national standard in Canada. Can we get everybody to adopt flow cross-matching as a way of getting standardized assessment of what the antibodies were in people's bloodstreams and documenting that? And so the Canadian Council for Donation and Transplantation, we presented to them, we asked for them to sponsor a workshop that we put on and hosted in Montreal. And what we were able to do is to actually get all the labs in Canada to standardize to using the same method. It took us 10 years to get there, but we got them all to standardize. And then we said, now with that standardization, we can actually develop a Canadian sharing system called the Kidney Paired Donation Program. So this is where you might have your sister wants to donate to you, but they're the wrong blood group, or you have an antibody against your sister. But you could have your sister donate to somebody else, and their donor could donate to you in a compatible manner. So that's the Kidney Paired Donation Program. That was launched in 2009. We've done over 1,100 transplants through that program. And then there's the highly sensitized patient program, where if you have a donor anywhere in the country and they match to you and you're highly sensitized, you get that kidney first from across the whole country. So that led to a Kidney Transplant Advisory Committee, the Kidney Paired Donation Program. We all work collectively through Canadian Blood Services, who took on the Canadian Blood Services is a company that's owned by the provinces. And so the provinces were willing to invest in CBS as an interprovincial agency for us facilitating sharing of organs between provinces. The new candidates were coming in. The high PRAs are the 99s to 100. You can see every cycle, match cycle, we would have more of these coming in. They would start collecting in the higher sensitization rate because they were really hard to match. And what we were seeing is that the 80 to 94% people were quite frequently getting transplanted, but the 97 and above were actually finding it hard to get a transplant through kidney paired donation. And then we launched in 2010 the highly sensitized program for patients with PRAs above 95%, meaning that they have antibodies that react to more than 95% of all donors that are available. And the first recipient, I was very happy about this, was a Manitoban. This individual here had blood transfusion. He'd waited, I think, 12 years to get an offer. And he got an offer from Halifax, a donor from Halifax. The kidney was shipped to Winnipeg, and he's done extremely well. This is all the active HSP candidates over time. And you can see that the PRAs of 100% are the ones that are accumulating. But the ones lower than that are actually getting transplanted through the program. When you look at it in terms of what's the approximate time to 90% getting match probability, you can see that 95 up to 98, actually 90% are getting transplanted by 9 months on the list nationally. Whereas many of these individuals had waited in their province for 16 years to get an offer. So this national program was shortening down their wait time to get access. Whereas if they'd just been in a provincial system, they would not have gotten access. And then the group that's still having difficulty is this 100% group. Only 50% are getting transplanted by 3 years while on this national list. So this is a group we're still struggling with. Again, it takes a whole team. This is the Canadian Blood Services team that supports the national registry that every day is in use. And so whenever there's an organ donor, the first thing they look at is whether there's a match in the national system for a highly sensitized patient. Okay, so where are we going? There's still lots of barriers. This is graft survival, patient survival, graft survival out to 2015. We still have acute rejection. We have lots of drug toxicities with the drugs that we have, and 40% of graft loss or death by 10 years after a deceased donor kidney transplant still is happening. And this is the patient perspective. This was Amy Silverstein, who I knew and was on Good Morning America and in the New York Times. And she's saying, you know, for 40 years, we have the same immunosuppressive drug combination. The drugs cause secondary disease, and in her case, she was developing cancer with her heart transplant, and she ended up dying. And her plea was, we need new, more effective drugs. And this is a really one of the key unmet needs is to come up with novel drug combinations to decrease toxicity while maintaining or enhancing the efficacy of the drugs that we have. So there's still lots to do in transplant. Other areas, optimize access through optimizing donor consent. So I think, again, policy things that we can do can still enhance consent rates. Alternative sources of organs, xenotransplants are the big topic now. I still think they're several years away. We're looking at, can we cross some of the memory with these highly sensitized individuals, and we're developing national policies to how to do this through the Canadian Blood Service system now. Optimizing donor quality to minimize ischemic injury, novel drug development, and then the final thing is around tailoring personal immunosuppression. I'm going to skip through that only because we're not going to have time. I do want to say that really this is where we have been. It's about immune risk assessment. We've learned a lot over the last 50 years. It's about trying to personalize immunosuppression, personalizing how we monitor the patients. We've been leaders in that here in Manitoba. And then optimizing patient outcomes. David Rush, who was my mentor here for many years, he graduated in Tucumán in Argentina. And the motto of their university was Pedes in terra ad sedera visus, which means feet on the ground and eyes to the sky. I think in medicine, we need to be grounded in biologic mechanism, but we need to be focused on the patient. And ultimately, the key is policy informed by both science and patient expectations. And if we marry these two things together, I think we can have very effective patient care, which is really where we're going, is patient-centered care in medicine. And with that, I'm just going to acknowledge that what I've been sharing with you is a lot of work over the last 30 years from multiple groups here in Manitoba, but really trying to show you how science and policy, when brought together, can actually be highly effective in delivering care for our patients. And thanks for your attention. So we have time for questions if there's questions, but I also know that speakers never finish early, right? So that's trying to adhere to the 50-minute rule. Yeah, go ahead.
I'm just wondering, I'm not sure if you mentioned it during this, but how do you determine the HLA next generation sequencing?
Yeah, so we have next-gen sequencing in the lab, and we will do that for the recipients. For living donors, we do next-gen sequencing as well. For deceased donors, we don't have next-gen sequencing available in the middle of the night. What we do is what's called a PCR technique. It's an SSO methodology, and then what we'll do is impute high resolution from that, but then subsequently do the high-res sequencing to confirm it. Jim.
Getting back to some science, where are we with the xeno to the pigs? Is there advancements being made?
Yeah, so the issue with xeno to pig is a couple of issues. One is there's a huge barrier that relates to galactosamine residues on the endothelium of pig vessels. And so what they ended up doing was knocking out a whole bunch of genes from the pig so that those proteins are no longer expressed. And they still have to come in with fairly heavy, intense immunosuppression to try and suppress that xeno response and then deal with the regular alloimmune response. The other problem with pigs are that pigs have viruses. And there's worry about zoonotic transmission of viruses in humans. So they've been knocking out a lot of the viral genes from the pigs as part of the development of these transgenic pigs. And so these pigs are actually kept in ultra clean facilities. They're doing multiple genetic modifications. They've done a handful of transplants, and most have not gone further than a few months, largely because of either rejection or infection, because of the degree of immunosuppression that we're using. So I think we're still a ways away.
Just a quick follow-up. Years back, Randy Ascoff was here, and we were involved with cyclospore in the San Diego. And we found that we had to go to germ-free rabbits in Minnesota. And they were just clean as a whistle because the garden variety rabbit just didn't work at all.
Right.
So what's the cost? I mean, has anybody looked at that and having
Yeah, so I think getting germ-free pigs is going to be a big order, really. And I don't think we're ultimately going to get totally germ-free. What they're going to try and do is knock out the key viruses that are integrated in the pig genome to be able to get you across those barriers. Yeah. Well, thanks everyone for your attention. I hope that was sort of illustrating a little bit about policy and science. Thank you.